products and some of the excretory organs. So without further ado, let's begin our topic today. Now, first of all, when we talk about excretion, the first thing that comes in mind is that excretory system is meant for removing the metabolic waste from the body. Now, this is just one part of the excretory system. There are many more important functions of the excretory system as well. So the animals, they depend on excretory system to maintain the volume, concentration and composition of their extracellular fluids. In our case, the extracellular fluids would be the fluid in our tissues, that is the interstitial fluid, as well as blood and lymph. Those are also extracellular fluids. And of course, not to be forgotten, to excrete the various waste. So the solute concentration of the extracellular fluid is very, very important, because if this concentration is less than the cytoplasm of the cell, what would happen? Water moves into the cells, causing them to swell and possibly burst. Because after all, animal cells don't have a cell wall. We just have the cell membrane, and if more fluid fills into our cells, the cell would burst. So that is why it is very important that the amount of solvent, that is water, which is present outside the cell has to be maintained in such a way that the cell integrity is also maintained. And in simple words, we can understand that blood, tears, sweat, these are also fluids, and they also have solutes, lot of salt in them along with water. And urine also has that. So the excretory system is balancing the amount of water and salts, and if these are in excess, those amounts will also be removed into the urine. So in this case, the solute concentration, we had seen if it is less, then what happens? If it is more, then what would happen? The water from the cell will move out and the cell would shrink. And if the cell shrinks, certainly the functions of the cells cannot be performed. So the solute concentration of the extracellular fluid affects both the volume and solute concentration of the cells. So both of these are interrelated. Now the physiologists, they use the term osmolarity in discussing osmosis. Now osmosis you have studied in various other topics, that it is a special type of diffusion in which we talk about the movement of water. Now to better understand osmosis, before we define osmolarity, let us take an understanding here. So here we have a solution in which there are two parts. One is the hypotonic solution and the other is the hypertonic solution, and these are separated by a semi-permeable membrane. So on this side you can see there is water, but the solute molecules are less. So this is a dilute solution and this is hypotonic. And here there are more solute concentration. So this is a hypertonic solution. And in the middle there is a semi-permeable membrane like our cell membrane. So water would move from where to where? That is, water is more in this dilute solution, in the hypotonic solution. So water will move from hypotonic solution to the hypertonic solution. So the net movement of the water will be more from hypotonic to hypertonic, and this movement will continue till the two solutions become isotonic. That means the concentration of water in both the solutions is at equilibrium. So this is what is osmosis, and it is a very common phenomena because the movement of water into and out of the cell takes place through osmosis. Now coming on to osmolarity. The osmolarity of a solution is the number of moles of osmotically active solutes per liter of the solvent. And here solvent, of course, as we said in the living world, is water. This definition is very similar, which you might have studied in chemistry, of molarity. That is the amount of or number of moles of solutes dissolved per liter of solution. Now here, however, when we say osmolarity, and here we are talking about osmotically active solutes. So let us take the example. If we say there is 1 molar solution of glucose, so it is also 1 osmolar, that is the value given as 1 osmol per liter solution. But when there is 1 molar solution of sodium chloride, then it is 2 osmolar solution. Why? Because in the water, sodium chloride will dissociate into 2 osmotically active ions, that is sodium and chloride. So that is why the osmolarity of Na will be higher even though both of them are 1 molar solutions. So this is what we have to keep in mind all through this chapter. Now taking an example regarding human blood, and this is more or less in most of the animals, the osmolarity of the human blood is 300 milliosmoles per liter. In short form, it can be written as MOSM per liter, whereas that of seawater is 1000 milliosmol per liter. So you can easily compare that seawater is having very high osmolarity. So that is why if our cell is put in seawater, what is going to happen? It is going to shrink because water from inside the cell will move out. So there is going to be exoosmosis. Now this part is of course very important in maintaining the integrity. So excretory systems, they control the osmolarity and composition of the extracellular fluid by excreting solutes that are present in excess and conserving solutes that are valuable or in short supply. So during the kidney function, we'll be studying that when the blood is filtered, along with the blood, because in the plasma, glucose, amino acids, NaCl and other salts are present. So out of that, which are very valuable for the body, those will be reabsorbed back into the blood, while which are in excess will be passed out in urine. So this way, kidneys are maintaining the osmolarity of our blood. Now excretory systems of course also eliminate the toxic waste products of the protein metabolism, and not only protein but also nucleic acids metabolism. The output of the excretory system is called urine. Now, first part of the topic that we are going to take up is osmoregulation, and what are the various osmoregulatory challenges that an animal has to face. So an animal can maintain water balance in 2 ways. One is to be an osmoconformer, and osmoconformer is that this type of animal will maintain its blood osmolarity same as that of the surrounding. So we can use the word isoosmotic or isotonic. Here this would mean the same. So here the blood or the cell cytoplasm is isotonic to the surrounding. And the other way is to become an osmoregulator. Now these animals, they control internal osmolarity at a certain value independent of that of the external environment. So their osmolarity is not going to be changed. While conformation means that these animals will change their osmolarity according to the medium. So if we put this entire thing in a graph here, and on x-axis we have taken the external osmolarity, and here on the y-axis it is the internal osmolarity. Now here you can see one is a straight line like this, rising. That means as the external osmolarity is changing, so is the internal osmolarity. Now this would be a case of conformation. So this is the way the organism is going to conform. While here we have constant internal osmolarity irrespective of the external osmolarity changing. So this would be regulation. So here, taking each type one by one. So first of all, starting with conformers. Now all osmoconformers are marine animals, but all marine animals are not osmoconformers. The reverse is not true. Now, these osmoconformers, their internal osmolarity is the same as that of the environment, so they don't have any osmoregulatory challenge. That means neither do they gain or lose ions, nor do they gain or lose water. So they don't have to expend any energy in maintaining their osmolarity of the body volume. And examples of osmoconformers are marine invertebrates, and among vertebrates there is one single example, and that is myxine or hagfish. Now this is a very primitive fish. Rather, among vertebrates we can say that it is the most primitive living vertebrate, and this is the one which is having its blood isotonic to that of seawater. So these are the examples. But for these animals, they will have to restrict themselves in the marine environment. They cannot move to other habitats, because in the other habitats there would be quite a lot of difference in the salinities, and with that case, like if we go into fresh water, the osmolarity in the fresh water is just 50 milliosmol per liter. So it is not possible to have the cell cytoplasm at that low osmolarity. Now these osmoregulators, they are able to live in environments that are otherwise uninhabitable for conformers, and so these regulators can live in freshwater, terrestrial habitats, and also they can move between marine and freshwater environments. So these osmoregulators, if they are in a hypoosmotic environment, so they'll have to discharge excess water, and if they are in a hyperosmotic environment, so in that case, the osmoregulator will have to take in water. So here they have to expend energy in maintaining. So whenever there is regulation, there has to be expenditure of energy. So who are the various osmoregulators? The vertebrates, except myxine, which we have mentioned that it's the osmoconformer, the only vertebrate which is a conformer. The other vertebrates are osmoregulators. So how do these osmoregulators maintain water and solute balance in their body? Let us now discuss that.